Q.1 Q7 Multiple choice question, MCQ, carry 1 mark each, for each wrong answer, 1/3. Q.1 Wheat plants treated with prolonged cold temperature at the seedling stage flower earlier than the untreated control. Seeds collected from these treated individuals, however, give rise to plants that do not flower early. This phenomenon is called A. Vernalization, B. Temperature acclimation, C. Photoperiodism, D. Adaptation. Q.2 Which one of the following plant taxa contains vascular tissue, xylem and phloem, but not woody tissue? A. Oak, B. Moss, C. Pine, D. Fern. Fern. Q.3 Which one of the following statements regarding spores and gametes is correct? A. Spores can directly undergo mitosis whereas gametes cannot. B. Gametes can directly undergo mitosis whereas spores cannot. C. Neither spores nor gametes can directly undergo mitosis. D. Both spores and gametes can directly undergo mitosis. Mitosis. Q.4 Which one of the following organelles controls gravitropism in the roots of higher plants? A. Chromoplast, B. Amyloplast, C. Chloroplast, D. Etioplast. Etioplast. Q.5 Phytoalexins play important role in plant defense against pathogens. Choose the incorrect option related to phytoalexins. A. Phytoalexins belong to secondary metabolites. B. Phytoalexins have antifungal activity. C. Phytoalexins are abundant in plants under normal condition. D. Different hosts produce phyalexins of varying chemical nature. Q.6 The figure shows the germination percentage of imbibed seeds treated with the given sequence of red R and far red F light, each exposure lasting 5 minutes. The percentage of germination was scored after 72 hours in darkness at 25 degrees Celsius. Based on this, which one of the following options is correct? A. Red light induces seed germination whereas far red light inhibits it. B. Red light inhibits seed germination whereas far red light induces it. C. Both red and far red light inhibit seed germination. D. Both red and far red light induce seed germination. N. H. CH. 2. COOH. C. C. H. 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 O. 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 COOH. The structures of 4 plant hormones are shown. Identify the correct hormone that is responsible for bending of coleoptile of canary grass in response to unidirectional white light. Q.8 Q9 Multiple select question, MSQ, carry 1 mark each, no negative marks. Q.8 Which of the following cellular components are not parts of cytoskeleton in angiosperms? A. Microtubules. B. Microfilaments. C. Intermediate filaments. D. Centrioles. Q. 9. Which of the following enzymes, when overexpressed, would result in rice grains with increased beta-carotene content? A. Phytene synthase. B. Carotene desaturase. C. Beta-glucuronidase. D. ENOLPYRUVALSHIKIMATE3 phosphate synthase EPSPS. EPSPS. Q.10 numerical answer type NAT carry 1 mark, no negative marks. Q.10. In spiral phyllotaxis, leaves are initiated sequentially on the meristem with 2 successive primidia being separated by golden angle. If a plant follows right-handed spiral phyllotaxis when looked down the meristem, then the angle between 2 successive leaves would be blank _ degrees with correct sign, round off to 1 decimal place. Q.11 Q15 multiple choice question, MCQ carry 2 marks each, for each wrong answer, 2/3. Q. 1. Match the cell tissue types in Group I with their corresponding total DNA content in Group II of a typical diploid angiosperm species and choose the correct option. C denotes DNA content in haploid genome. Group I Group II. P. Pollen tube 1, 1C. Q. Megaspore mother cell 2, 2C. R. Synegid 3, 3C. S. Embryo sac prior to fertilization 4, 4C. 5, 8C. A. P-1, Q2, R3, S5. B. P-2, Q2, R1, S5. C. P-1, Q2, R1, S5. D. P-2, Q1, R2, S4. Q. 12. Match the modified organs in Group I with their corresponding prototypic forms in Group II and choose the correct option. Group I Group II. P. Tendrils in grapevine 1. Modified stem. Q. Tendrils in garden P2. Modified leaf. R. Spines. S. Thorns. A. P-1, Q2, R2, S1. B. P-2, Q1, R1, S2. C. P-1, Q2, R1, S2. D. P-1, Q1, R2, S1. Whirl 1. Shea. Pal. Whirl 2. Petal. Whirl 3. Stamen. Whirl 4. Carpel. A. C. A. C. A. C. A. C. Q. (13). The diagram describes the ABC model of flower patterning in Arabidopsis where the A, B and C functions are operational in the whorls 1 plus 2, 2 plus 3, and 3 plus 4 respectively in the wild-type flower. Removal of A or C function results in the floral organ arrangements as carpel, stamen, stamen, carpel or sepal, petal, petal, sepal respectively. Based on these observations, which one of the following molecular pathways is correct for floral organ pattern generation? Arrow indicates activation and bar indicates inhibition. A, B, C, D. Q. 14. Find the correct match among the plant species in Group I, the predominant phytochemical in Group II and the economic medical use in Group III. Group I Group II Group III. P. Syzygium aromatica Mi, vincristine 1. Toothache relief. Q. Gracilaria sp. E. Eugenol 2. Dessert jelly. R. Catharanthus roseus E. Agar 3. Leukemia treatment. S. Theobroma cacao IV. Morphine 4. Analgesic. T. Papaver somniferum versus flavanols 5. Beverage. A. PI1, QE3, RE4, SV5, TIV2. B. PE1, QE2, RI3, SV5, TIV4. C. PE1, QE2, RV4, SI3, TIV5. D. PI2, QE3, RIV1, SE5, TV4. V4. Q. 15. Match the genetically modified crop in Group I with the corresponding genetic element in Group 2. Group I. Group 2. P. Tomato with delayed fruit ripening 1. EPSP synthase. Q. Herbicide-resistant soybean 2. 12-desaturase. R. Insect-resistant cotton 3. Polygalacturonase. S. Soybean with modified oil content 4. Bt-cry protein. A. P3, Q1, R4, S2. B. P-1, Q3, R2, S4. C. P-2, Q1, R4, S3. D. P3, Q2, R4, S1. Q.16 Q18 Multiple select question, MSQ, carry 2 mark each, no negative marks. Q.16. To understand the mechanism of systemic acquired resistance (SAR), a team of researchers isolated a mutant with reduced SAR response. Sequencing of this mutant revealed homozygous mutations in 2 genes, X and Y. Which of the following experiments would test whether the mutant phenotype is caused by mutation in either or both the genes? A. Complement the mutant with X or Y and analyze the phenotype in each case. B. Complement the mutant with both X and Y and analyze the phenotype. C. Cross the mutant with wild type and analyze the segregation pattern of the phenotype. D. Compare the expression of X and Y in mutant and wild type plants. Q.17. The observations of an experiment on seed germination in various genotypes under different light conditions are given, where square root and X indicate germination and the lack of it, respectively. Genotype: blue, red, far red, white, dark. Wild type: square root, square root, square root, square root, X. Cry1 mutant, X. Square root, square root, square root, X. Fear mutant, square root, X. Square root, X. PHYB mutant, square root, X. Square root, X. VP1 mutant, square root, square root, square root, square root, square root. Based on these observations, which of the following options is correct? A. All the 3 light qualities blue, red and far red are required for seed germination. B. Any one of the 3 light qualities, blue, red and far red, is sufficient to induce seed germination. C. The CRY1, FEAR and PHYB proteins are required for blue, red and far red light perception, respectively. D. The VP1 protein is unlikely to be involved in light perception. Q.18. Which of the following options is correct in the context of hybrid plant generation using Barnase-Barstar-based male sterile lines? A. Barnase inhibits Barstar. B. Barstar inhibits Barnase. C. Barnase and bar star are used to generate the male sterile line and the restorer line, respectively. D. Barnase and bar star are used to generate the restorer line and the male sterile line, respectively. Q.19 Q20 Numerical answer type NAT carry 2 mark each, no negative marks. Q.19 In a diploid plant species, the T allele produces tall individuals and is completely dominant over the T allele that produces short individuals. Similarly, the W allele produces round seeds and is completely dominant over the W allele that produces wrinkled seeds. Assume T and W loci not linked. If a parent with TTWW genotype is crossed to another parent with TTWW genotype, the fraction of the F2 population produced by the fusion of both recombinant gametes would be blank. Round off to 2 decimal places. Q.20